Hi everyone. From this video onwards, we are going to learn about the microscopy topic, in which we will discuss about the history of microscope, then types of microscope, structure and principles of microscope in the different consecutive videos. In this part 1 or the first video, we are going to learn about introduction to the microscopy topic. So here, coming to the history, the most important tool of biologists is the microscope, and the invention of light microscope in the 16th century is perhaps the single most important contribution ever made to the advancement of biology. And the microscope is going to be the combination of two words where we call it as a micro means small and the scope means a view. And the optical microscopes are going to be considered as the oldest and simplest of the microscope. So whatever the present day microscope we are using in the biological laboratories has been developed over the course of several centuries. That means it was not made instantly. So it took many years to come into the advanced type of microscope that we are using at present. Ancient Romans, Greeks and Indians had known lens of glass and quartz. In the 14th century itself, spectacles and the single lens were used as a magnifiers, and the Zacharias Janssen, a Dutch astronomer, added one more lens and constructed a true telescope. And then Mr. Campini, an Italian, also found that a lens can be ground to the desired curvature. Since the curvature of the lens increases its magnifying power, microscope thereafter became a very powerful tool in the hands of biologists. Then coming to Mr. What we call as Robert Hooke, he was one of the first scientists to actually observe the microbes in 1665, and his illustrations and the observations from a variety of objects viewed under a microscope, and it was published in the book called as Micrographia. And then coming to Mr. Antony von Leeuwenhoek and the duration of Antony von Leeuwenhoek, we know that it is 1632 to 1723, and sometimes wrongly created with the invention of the microscope, who used his single lens. So here, this is a microscope that was used by Mr. Antony von Leeuwenhoek to observe several minute objects like insects, mold, then pond water, saliva, etc. Okay, and he was the one who made the handmade microscope with a single lens. And then coming to the terminology, so here we are supposed to learn about few terminologies related to the microscopy topic. So here comes the microbiology. It is a branch of science that deals with the study of microorganisms that we know. And then what are microorganisms? Which cannot be seen with the naked eye and can be seen only with the help of the microscope, are going to be considered as microorganisms. And then what is a microscope? So this microscope is an optical instrument which is used to observe the microorganisms, which is very important for the biological laboratories. And then what is meant by microscopy? The study about the microorganisms using microscope is going to be called as microscopy. The study about the microorganisms using microscope is going to be called as microscopy. Then moving to the actual definition of a microscope. A microscope can be defined as an optical instrument consisting of lens or a combination of lens for magnifying the image of minute objects. Not only that, it also should have a good resolving power or resolution. So we will discuss what is this resolution and the magnification in the consecutive video under the topic called as principle of microscope. Just remember, a microscope is going to be considered as an optical instrument. What is meant by optical? Possessing the lens or a combination of lens which are used for magnifying, that is enlarging the image of the minute object such that we can see, and it also going to possess a resolving power. That means clarity. So resolving power means, simply in terminology we'll call it as a clarity. So if a particular optical instrument having these two properties, that is magnification and the resolution or resolving power, then we are going to call that as a microscope and mainly used for observing the microorganisms. Then moving to the working of a microscope, so we will discuss in detail about it, but here have a glance of it. So the main function of any microscope is to produce the large and clear image of the object such that we can see them. And not only that, I told you that clarity is often referred to as the resolving power of the microscope. And in order to understand the functioning of a microscope, it will be necessary to understand as to how our own eye resolves and produces an image of an object. Here, if you see, the lens of the eye is going to focus the light reflected by an object and projects an image on the, that means in our retina or on our retina, so that is retina, retina. Now this retina is a granular matrix consisting of light sensitive cells called as cones as well as the rods. That means our eye possesses the cones and the rods. Now, these cells, that means whatever the retina and the rods, are going to convert the light impulses falling on what we call as nerve impulses. So, whatever the light impulses are going to fall on the retina, that is cones and the rods position, they are converting that into the nerve impulses, producing a sensation of vision in the brain. Now the rods and the cones are placed 5 microns apart. That means the difference between the rods and the cones is going to be 5 microns apart. Hence, the eye cannot resolve details of an object less than 5 microns in size. So that is very important. So if the size of any object is going to be less than 5 microns, we cannot see. Why we are unable to see? Because the rods and the cones are placed 5 microns apart. When an object is brought closer to the eye, it grows larger. But when it is brought too close, that means even near to the eye, then the lens of the eye cannot focus the image and the object appears blurred. And the shortest viewing distance of an human eye is going to be of about 25 centimeters. At this distance, the limit of resolution, that means clarity, is about 0.2 mm. And the objects lying together closer than 0.2 mm will not be seen separately by the human eye. So this is the basic working of a microscope with considering with the eye. Then moving to the brief note of the types of microscopes. So here you can see we are going to have the 2 types of microscope depending upon the magnification principle involved. One is going to be called as a light microscope that we generally use in our laboratories at the school level and the college level. And the second type is going to be the electron microscope, where the specific high institutes are going to possess this electron microscopes like a CCMB, like that. Then moving to the light microscopes. So the light microscopes are the one which are going to use the light as the illumination source. So there it is going to be of 2 types. That means depending upon the light, we are going to have the classification of the 2 types of the microscope. One is going to be called as a simple microscope, where only one biconvex lens is going to be used, and the second type is going to be the compound microscope, where consists of more than 2 lenses, or at least 2 lenses should be there in the microscope. Then we consider them as the compound microscope. So at the school level and at the college level, +1, +2 level, these are the microscopes that we are seeing in our biological laboratories, isn't it? And the compound microscope magnification is going to range from 1000 to 2000 times the diameter of the specimen. And the wavelength of the light that we are using in this light microscopy is 400 to 700 nanometers, which is nothing but the wavelength of the visible light range. And most commonly used compound microscope that we are using to study or bright field microscope, dark field microscope, then phase contrast fluorescence. Along with that, we are also going to have the ultraviolet microscope, interference microscope like this, but majorly we'll discuss these four microscopes under the compound or light microscopes. Then coming to the electron microscope, this electron microscope was developed by Knoll and Ruska. And these electron microscopes, instead of the light, they use a beam of electrons as a source of illumination. And instead of the lens, we are going to have the magnetic field. So that's the difference between the light microscope and the electron microscope, where there will be no usage of any lens or the what we call as a light. So instead of those two, the source of illumination is beam of electrons, and the instead of the optical lens, we are using the magnetic fields, and the resolving power is very high when compared to the light microscope, and that's the reason why we are able to see the viruses under this one and the internal structures of cells. Okay, and the magnification power you can see much higher, where in the light microscope it is only about 1000 to 2000 times, but here you are going to see sort of 1 lakh times of an electron microscope is much more higher than light microscope. Then we are having the two types of electron microscope. One is a transmission electron microscope, and the second one is going to be the scanning electron microscope. So we will discuss all these topics in detail in the consecutive videos. Here just I am introducing these all terminologies to have some sort of a basic knowledge about it. Then moving to the uses or applications of any microscope, it may be electron or whatever it may be. So the first point that we are going to discuss is microscope is very useful for students in schools and colleges for conducting their academic experiments, that we know very well. And this microscope is going to be of a very immense help, as the students can now see the bacteria and viruses which is otherwise invisible to the naked eye. Thereby, they witness things which they have studied in the theory. So that's how the microscope is very useful. And coming to the plant cells and the microorganism living on them can be observed under the microscope. So a compound microscope has proved beneficial to biologists too. And microscope is of great use in the pathology labs as to identify the disease. That means, for example, if you consider the malaria can be diagnosed with the help of the microscope, where the RBC cells possessing the life cycle of this plasmodium can be identified. Then in forensic laboratories, microscopes are used to identify presence of minerals or metals in the human cells so as to solve the criminal cases. And the forensic experts can also find out the origin of a drug by viewing its component particles under a microscope. So this is all about the brief introduction to the microscopy topic. From the next video onwards, we are going to discuss about the structure of the microscope. Then we are going to discuss about the principle of the microscope. Then we are going to discuss the different types of the microscope, that is light microscope types, as well as the electron microscope types in the different consecutive videos. Thank you.